Hi, welcome to my talk. So my name is Joel Pearson. I'm a professor of cognitive neuroscience here at the University of New South Wales in Sydney, Australia. And I'm going to be talking about measuring aphantasia, measuring mental imagery and its impact. Its impact on the way we think, its impact on our cognition, and a few other things. So let's get started. I wanted to start off this talk by just mapping things out a little bit, the way I like to see things, the way I like to think about measuring the mind's eye, measuring the mind. And it's this kind of graphical equation, if you like. One side here, we've got a wireframe image of my brain, and on the other side here, the mind. And in sort of neuroscience, we hear a lot and all the time of things happening with the brain, right? We hear these huge breakthroughs of hacking neurons to get viruses in there, to get neurons to respond to light, making tissues clear, and stuff we're doing in the lab with complex algorithms and brain scans, looking at complex spatial dynamics of the brain, all on this side of the equation, like looking at the wetware of the brain, how it works. Really cool stuff. It gets a lot of attention, a lot of funding. On the other side of the equation here, the mind, we don't hear as much, right? We tend to rely on the classic methods, things that have been around for a long time to try and measure the mind. And of course, ultimately, what people are doing with the other side of the equation is really to try and understand the mind as well. And those two things, at least with humans, are intertwined in a way that we don't want to pull them apart. So my point is that too much of the time this other side of the equation is not sexy enough. It's not as exciting, and it gets left behind a little bit. So what we've tried to do here in my lab, Future Minds Lab in Sydney, Australia, is try and figure out better ways of measuring the mind, ways of trying to make that objective, make it reliable. So it's not just people, not just us talking about our memories and our opinions about how we experience the world, right? So trying to, if you like, get a microscope for the mind, trying to develop that blood test for the mind, trying to develop more sort of objective, hard methods if you like, but that's not a great name, I don't like that, like we have here for the brain, but for the mind. So that's what I'm going to try and convince you of a little bit today, right? That we can have a microscope to try and measure dimensions of the mind, and of course, in particular, mental imagery, the imagination, therefore, aphantasia. So the way we first figured out how to measure mental imagery, visualization capacity, if you like, was hacking or using a type of stimuli, ambiguous. These are ambiguous bistable stimuli. You've got the young, old woman, the classic faces and the vase there, or what's called this wireframe Necker cube. And these are interesting because you can look at them for a long time and the way you see them, your interpretation changes back and forward. It alternates over time. And I'll just keep doing that over and over and over. And one of the most exciting, the coolest version of that is something called binocular rivalry. It's something I played around with a lot during my PhD, which seemed like a long time ago now. So what is binocular rivalry? Well, the sort of easiest way to get binocular rivalry is if you just put up a hand in front of one of your eyes, so one of your eyes is looking at your hand, and the other eye is looking at the monitor, looking at me or anything else in the room. Right, so you're literally putting your visual system into a rivalry situation. The information coming in with two eyes is battling it out. One eye is trying to see your hand, and the other maybe is trying to see your computer monitor. Right, so that is binocular rivalry. And if you can equate those two images, pretty hard to do just by looking at it with lighting in the screen right now. But if you can keep them equal, something really interesting happens. Rather than seeing sort of this ghosting, sort of transparent hand, which you're probably seeing now, we see something different. We see that your brain solves this rivalry with these beautiful oscillations or vacillations in consciousness, right? The way we tend to do this in the lab is with very different patterns, a red and a green one we tend to use with different stripy patterns or orientations. So they're very different. The more different, the better the rivalry. And so this was a fascinating way to study consciousness or awareness in itself. But the reason I'm talking about this today is that it turns out you can use this illusion, this binocular rivalry illusion, for something else, not just to study consciousness, but to measure visual imagery, to measure the imagination, if you like. So this is how the paradigm works. We first we get people to come into the lab, they sit down in a dark room, and we're going to use a binocular rivalry stimulus like you just saw, red and green. And we're going to get people to first imagine one of those two patterns. There's a really faint green pattern there in this diagram. So let's say I'm sitting down, I'm imagining the green pattern, then I stop doing that, right? I have a rest, I do nothing, or I do a filler task. There's lots of different versions. And then all we do is flash on that binocular rivalry stimulus. On, off, very brief, okay? It's not staying on, so we're not seeing those oscillations, those beautiful vacillations in consciousness. It's just on and off. And all the individual in the test has to do is tell us which pattern they saw, the red or green. So something really interesting, really cool happens when we do this is that the pattern you imagine tends to influence what you see in binocular rivalry, right? So you imagine the red pattern in this animation, we flash on the binocular rivalry, you see red. You imagine the green, you're more likely to see the green in binocular rivalry, right? So there's this bias, the content of what's in your imagery, what's in your imagination is biasing this visual illusion. And this is really interesting for a couple of reasons. One, it's kind of cool that things I can just come up with in my mind will literally change how I see the world. That's pretty cool in itself. But two, we realized that we could use this as a way to measure a dimension of mental imagery, of visual imagery, right? And there's lots of different ways of doing this. The way we tend to do it is, right, we've got a red and a green pattern in this example, and so it's 50-50 chance, two patterns, right? And any bias I get in the way my imagery can affect how I'm seeing binocular rivalry, we can think of as a bias here above chance. So that's how we tend to do that in an experiment. We look at that, how much difference from chance there is, and that is a measure of visual imagery strength. And there's good reasons why we call it imagery strength. You hear a lot of people talking about vividness of imagery, but I like to think of all the other dimensions of imagery. And at least functionally, imagery tends to look like and behave in our experiments like a weak version of normal visual perception. Doesn't mean they're the same thing. I don't think they're the same thing. There's good evidence that they differ in a number of different ways, but at least functionally, the data looks very similar between imagery and weak perception. That's if you have imagery, of course. Right? So that's why you'll hear me and people from my lab, or you read our papers, talking about the sensory strength of mental imagery rather than often other dimensions like vividness. Right, so as I said, imagining things changes how we see the world, which is pretty cool. And using this method, we can measure the sensory strength of mental imagery. So why is this a blood test? Why is this a microscope to measure the mind? Because we don't have to rely on people telling us directly, verbally, trying to interpret their own mental images. In many of the questionnaires, you have to interpret your own mental image and give me a number for vividness, for example. In this method, you don't have to. You don't have to say anything about your imagery. All you have to do is tell us about the binocular rivalry display. And we even have versions of this that we've published where you don't even have to do that. We can put a little probe in that binocular rivalry display and therefore take out the need to tell us about that binocular rivalry. But that's a bit beyond today's discussion, right? So that's kind of the first method I like to think of as a microscope, or a first step towards objectively measuring the imagination, objectively measuring visualization capacity or strength. So what can this tell us about our aphantasia? So we use this method for trying to see if we could, I'm not going to say diagnose because I don't like that, it has a negative connotation, but so we can sort of assess the validity of aphantasia using this sensory measure. Right, so we got people into the lab here and we got them sit down in a dark room and we use this binocular rivalry method to measure their sensory imagery strength or lack of it. So here's some data, and this is from our original, in the grey there, 2018 paper. And we see a distribution there, but that's centered, the mean is around 50%, right? Again, two patterns, 50% chance. And so on average, we don't see for that group any priming above chance. When we look at a whole lot of people from the general population in the red there, or pink, then we see priming right up to 100% down to around chance again. And you can even see, actually interestingly, there's sort of one or two individuals who said they had aphantasia who were getting some priming. And that's an interesting scenario there to try and interpret what that means, why there's priming there if people are identifying as being aphantasic. Here's some sort of update on that data there in the turquoisey color, and that's all the individuals with aphantasia we've measured using this binocular rivalry method so far. And again, the mean is still sitting there around 50%. And just here you can see a histogram that's showing the normal population here, the distribution around to 60%, I think. And you'll see that our original 2018 paper using this method is here, and then all the data we've tested is just slightly above that. So using this more objective method, measuring the sensory strength of mental imagery, it does seem that those who, on the whole, identify with as being aphantasic aren't getting this priming. Their imagery or their attempts at imagery are not affecting the binocular rivalry stimulus. So in other words, it doesn't seem that there's some kind of unconscious imagery there that those individuals are just not aware of. There's nothing there, or nothing to the degree that can affect subsequent binocular rivalry at least. So another method we developed, I guess, in the lab to try and tap in or measure aphantasia itself or the effects of it is to try and use the emotional response. So there's some interesting literature in clinical psychology suggesting that mental imagery plays this really important role, but sort of linking our thoughts and our emotions. And by sort of, if you like, if imagery is there, it can make some dimensions of our thoughts almost seem real and therefore fire up our emotions in a much stronger way than thoughts without the sensory component. So we designed this experiment. We got people into the lab and we wire up their fingers with these little electrodes, and that's going to measure their skin conductance response. So that's basically how much they sweat, right? It runs a little current between those two fingers. You can't feel anything. It's so faint you don't notice a thing. But it's basically measuring, how much we sweat. And when you see something emotional or hear a scary story, you sweat ever so slightly more because your brain is responding in an emotional manner to that stimulus or that story. So this is our objective physiological measure of the emotional response. So we set people up like that. They come into the lab, sit down in a dark room looking at the computer monitor. Then they have to read these scary stories we designed. And they're things like this. You're in the water. The sun glints off the waves. Suddenly a dark flash in the distant waves. Maybe it's a shadow. You turn to the beach. People are pointing. People are shouting and screaming. A large fin slices through the surface of the water, moving closer and closer. A mouth opens wide. Your legs flail. White teeth. Right. So if you live in Sydney, Australia here, and you swim in the beaches, this kind of story is pretty scary because in somewhere in the back of your mind, you're always sort of picturing a Jaws scenario or some kind of shark. Anyway, there's not just one story. There's rock climbing. There's lots of different stories that are all kind of scary in a sense. So here's what this skin conductance response looks like for what I'm calling controls, those with mental imagery. You get this nice monotonic continuing response as the story goes on. This is the time the story's on the screen. It gets stronger and stronger. So here's the question, what's going to happen if you don't have any mental imagery? We see here the aphantasia data in the red, and we see basically a flat line. We just don't see this monotonic response to the emotion. And there the means there of those two. So that's interesting, right? It's telling us that at least suggesting that the emotional response to these stories is different in these two groups. Just from those data alone, we don't know whether this is about general emotion or it's about imagery specifically. So we need to run a control condition. So we run a similar experiment, basically the same thing, but rather than having the stories on the screen, now we're showing yucky, scary, or emotional images, right? So there's no need to conjure up something in your mental imagery. Now the things are there pictured on the screen, right? So here's the idea. When we run this, is there still going to be a difference, or those two lines, the blue and the red lines, going to be the same? And they're basically the same. Not exactly, but more or less. You can't distinguish those with the statistics. Right, so these perceptual images on the screen get an emotional response in both these groups, right? So this is really suggesting it's not anything to do with emotion. Emotional responses seem the same if you have imagery or no imagery. It's about the emotional response to just the written word or just the story, right, when there's no images there for your eyes to see. So it seems to be a role for mental imagery. And as I said, this plugs into clinical theories of the role of mental imagery. So that's sort of, I guess, number two, our second method, that we can use the emotional response to emotional stories that require imagery to amplify that emotion. And when imagery is not there, we see a much lesser emotional response, right? So that's, again, objective, physiological way of measuring imagery and therefore aphantasia. So that's the second. So we have two methods so far, the binocular rivalry and this emotional story one. Next, I want to talk about something to do with the eyes, right? Can we use the eyes and indeed the pupil response, how wide our pupil is, to try and measure imagery and therefore aphantasia? So, you know, if you look around the room right now, you look at a bright light, your pupil contracts, right? It's too bright. It's your eye trying to block out the light from damaging the cells at the back of your retina. So that physiological response happens without you having to do anything. It happens automatically, and it happens every time you look at bright light and you go into the dark again and your pupils open back up again. So it's not just bright light, even though it's called like the light response. There are other scenarios when you're thinking really hard, your pupils change, and there's evidence that when you imagine bright lights, your pupils might respond too. And some illusions and other things like that which seem to internally drive that pupil response. So we wondered if we could use this pupil response to try and measure imagery and then try and measure aphantasia, again as an objective physiological measure that was directly tapping into something in the brain. So we used a paradigm, here's a sort of timeline of it, from another study. And what we're doing is first showing people these little triangles and then getting them to imagine the triangle. Very simple task. And they can imagine or see a single triangle or multiple triangles, 4. And here's the catch. They can either have to imagine a dark triangle like this one here or a white triangle, a light triangle. And you can probably already guess how we're going to analyze the data is looking at the dark and the light and if there's a difference there. Right, so first you see it on the screen, then you have to imagine it. So what does that kind of data look like? So here's the time across the experiment. There's the perceptual part of the experiment. You've got the dark and the light condition there. The blue in the dark is quite different here. Again, the background's always the same. It's just that little triangle. So in the bright condition, when the bright triangle is white, the pupil is contracting. When the triangle is black, the pupil is much wider. Right, then we switch that off, things come back to normal. And here, when you have to imagine that triangle, what's going to happen? Here's the interesting question. And we see a nice difference. Let's zoom in there. There we go. A nice difference in those data, just like during perception. When you're imagining a dark triangle, your pupil is wider. And when you're imagining the bright triangle, it's contracted in comparison. Right, so it seems that even though there's nothing on the screen, the content is all in your mind, your pupil's still responding. So here's a question. We do the exact same experiment with people with aphantasia. What's going to happen? Well, here's the first part of that data timeline. And again, we see a nice separation in the perceptual condition. Right, so perceptually things are working fine, that the pupil's responding in the way it should, nothing out of the ordinary there. We switch that off in the dark background, things come back together. Here, then we go to the imagery period. And what we see there again is, not again, but we see there's no difference. Okay, so these individuals with aphantasia that we're testing, they're trying to imagine, or get to how we know they're trying to imagine in a second, but there's no difference between the dark and the bright condition there. So their imagery just can't differentially drive their pupil in the same way it can if you have imagery or attempts at imagery. Right, so it's pretty cool stuff. I think this is the first sign that we have this objective or third method to objectively quantify and measure aphantasia using your eye response to bright and dark imagery or imagery attempts. So like I said, how do we know that those with aphantasia are actually trying? And this is something that skeptics sort of come up with and say, oh, people, they're just saying they're aphantasic, they're just not trying to do the imagery, or it's psychogenic, they have imagery that's fine, for some psychological reason they're not doing it. I think these data speak most strongly to that. So I said before, right, we have a condition where there's one triangle and a condition where there's 4 triangles, so different set sizes. And we know from other literature and lots of other papers that when cognitive load is greater, right, when you're trying harder to do something, your pupil responds to that cognitive load, to effort, if you like. That's exactly what we're seeing here. So this is aphantasia data I'm showing here, and you have the data here is for set size 1, single triangle, here it's set size 4, so 4 triangles. And we see a significant difference in the pupil response across the 2 set sizes. So we know that when there's more triangles, those with aphantasia are trying harder because their pupil is responding differently, even though it's not doing that differentially in the dark or the light condition. So I think this is nice evidence that those people in our study, all the people in our study, were trying hard to conjure up a mental image. And it's only those with imagery who we saw this differential pupil response. So we also got people to report the vividness of their imagery during those, when they were imagining the triangles. And so we can map that vividness data onto the pupil response here. So here, the vividness ratings 1, 2, 3, or 4. And then we can look at the sort of difference in pupil diameter across all those different vividness ratings for set size 1, set size 4. And we see nice significant results there where the more vivid you're rating that single episode, or that single episode where you imagine a triangle or triangles, predicts the difference in pupil score, the difference between the light and dark. And the same for set size 4. So these physiological responses are related to the vividness of the imagery during that period. And likewise, we can do a similar thing with our sensory measure of imagery strength, this binocular rivalry technique. We can map on to the pupil data, right? And when we do that, some correlation graphs here, we have the pupil difference score here, and on the horizontal, the priming in the binocular rivalry task I just explained. And you see this nice significant prediction there, both set size 1 and set size 4, for those with imagery, not for those with aphantasia. So it seems that these pupil responses are related to not only the imagery vividness, but also the sensory strength, which is a nice way to triangulate a measure of imagery, I think, and a nice way to assess, again, I'm not going to use the word diagnose, assess the existence of aphantasia in a given individual. Right, so I think we have at least 3 different ways of measuring imagery and therefore measuring and assessing aphantasia. The binocular rivalry method, sensory strength, the lack of skin conductance response with scary stories, and this lack of pupil response in the eyes. So there's 3 different objective and, on the whole, more reliable ways of assessing imagery and therefore aphantasia. So I think we're moving the field, we're moving towards having these objective techniques and technologies, and we can now start to look really under the hood at what is aphantasia, what's causing it, where does it come from, and what are the implications and impact to your lives if you don't have imagery. So yeah, this is sort of our attempt, if you like, at getting a microscope for the mind, a blood test for the mind, assessing mental imagery objectively without having to rely on subjective self-reports, metacognitive self-reports. So I want to pivot now and talk about sort of another stream of research we've been doing around aphantasia, and rather looking at methodology and how to measure it and assess it, looking at the impacts it has to general cognition, to thinking, and a range of other aspects. All right, so is your mental life different with or without imagery? If you think in pictures, how is that different? How does it change the way you think? And therefore, how is it probably going to change your life? And Starting at the sort of basic level, again, I'm going to switch back to questionnaire data now. This is some data we have looking at trying to sort of get at the dimensions of aphantasia, whether they're purely visual or they go across all of the senses here. And so we're showing data here for people with imagery and then people who identify as being aphantasic. And you'll see a big dint here in the visual domain, but you'll see a massive difference here across all the senses. And we've got a lot of data on this now. So you can do all kinds of clustering analysis and all kinds of different ways of looking at grouping. And again, it depends. There's a whole lot of choices you can make here about how to cluster, how to group, how to define based on the questionnaire, just based on the data. But in all the different ways we've looked at, it's safe to say between 40 to 80% of the people we've measured now have what I'm going to call multisensory aphantasia. What does that mean? It means that people are reporting floor scores, no imagery in any of these senses: visual, auditory, tactile, kinesthetic, gustatory, olfactory, and emotion there in red. So at least 40%, and with some measurements or some clustering, up to 80% of people are falling into this category. And only a much smaller percent, between 8 and 10, or between 8 and 12% maybe, are what I'm going to call pure visual aphantasic. So it's purely visual aphantasia. And then there's a whole lot of groupings in between, which I'm not going to go into right now, but I just wanted to give you a flavor of these distributions. So I think it's fair to say that we can call this multisensory aphantasia. I know that people are trying to come up with different names for different types of aphantasia, but I think it's just simpler and clearer and easier for everyone if we just call it multisensory, visual, and if someone just has only auditory, then auditory aphantasia, etc. Just simpler and clearer, makes a lot more sense. No one has to memorize and learn new words. So dreams is another interesting thing that is a lot of questions around dreams. Do people with aphantasia dream? So here's data from a whole lot of aphantasic individuals. And again, in the reddish color is the aphantasics, and the blue, those with imagery. You'll see, particularly in the sensory domains of the dreams, right, which makes sense, you see some significant differences there. But on the whole, not too many differences. Important to say this is average data of a lot of people. When you start drilling in and clustering and looking, you see that some people report not dreaming, some people report dreaming pretty normally. Again, some interesting work to be done there. To really get at the bottom of this, you want to really do sleep studies with people in the lab, come in there, sleep, when they go into rapid eye movement, sleep, and you wake them up and ask them if they were dreaming. And lots of people who say they don't dream, turns out do dream when you wake them during that period. So I'm really fascinated not only in terms of voluntary imagery, but this relationship with involuntary imagery and hallucinations. So imagery like in the dreams, where you're not effortfully trying to create that image, it's just boom, there. So if you guys can have a look at something like this, you may have the faint experience of some redness, some red pixels, because you're looking at a screen, there's no ink, red pixels here in the center. And if I do this, then that redness goes away. Maybe it comes back. Maybe it goes away. So this is something called neon color spreading. It's a visual illusion, and it creates a hallucination of red or light. And a type of imagery I'm going to call involuntary imagery. You experience red in the center there where there is no red hitting your retina, hitting your eyes. So it is being created like a mental image, but it's being created automatically like in a dream. So we ran a study trying to assess, is this type of automatic involuntary imagery normal or not normal, but there in those with aphantasia, or is it different? So we set up an experiment like this where it's on a sort of a induced red square there, and the question is, you have this little slider thing, and you have to adjust this red square to match the red in the center there. And so if you've done this, maybe some of you listening have done this experiment and you might remember. So here's some data, and what we're seeing here, now I'm flipping the colors, the aphantasics are in blue and controls in red. And this is data for a dark background and a white background. And we're seeing that on the whole, and significantly so, those with imagery are reporting in both those conditions stronger involuntary imagery, stronger red or green, we do this with different colors, in the center there. And we can also ask people to subjectively give a rating, more subjective measure of that. So rather than that adjustment method there, here they're giving a rating, and we see again significant differences there. Although in both, a large distribution, which again is going to be one of the themes of aphantasia, and in fact for all of us, that looking at the means can sometimes be a little bit misleading, because you do have these big distributions and there is a lot of variability there and individual differences. But on the whole, when you look at the averages, there is a difference between those with imagery and without imagery in terms of these neon color illusions and also dreaming. So we also have a bunch of different experiments looking at looking at memory, short memory, long-term memory, episodic memory. And we've been studying short-term, what's called working memory in the lab for a number of years now. And in particular, looking at the relationship between visual working memory and visual mental imagery. As these two are sort of separated into two different fields, and we wanted to see if people are using their imagery as a tool, a mnemonic, to help them solve these memory tasks. So we got people into the lab. They came in here physically pre-COVID to test their working memory. Right, so we're doing visual working memory, and we do this a number of different ways. One way of doing this is with this little tiny, hopefully you can see that little tiny stripy patterns. And there could be 1 on the screen, there could be 3, 4, or 5, there could be sort of different numbers, and you're trying to remember them all, both where they are and their orientation. So that flashes on, that goes away, and there's a blank screen for 6 seconds, and then a single one of those will randomly flash up, and you have to say, was it rotated clockwise or counterclockwise compared to what was there previously, 6 seconds before. Right, fairly standard way to assess visual working memory. So here's some data, and that's right, you can't really see the two lines because they're on top of each other. So the blue is those with imagery in the general population, and then the grey little squares there, those with aphantasia. And this is set size across the horizontal. This is a classic finding. The more things you have to remember, the worse you do, right? Makes sense, right? You have to remember a really long phone number. You're not going to be able to do it. It gets too hard. A single number, very easy. Same thing with visual patterns, right? So you have this nice drop-off, and turns out there's no difference whether you have aphantasia or not. Even though, right, there's good evidence that if you have imagery, you use it for these memory tasks. Pretty interesting, right? So next, you can do the exact same experiment, and we did, rather than visual working memory, we look at sort of number or spatial number working memory. It's the exact same deal, but now we have little numbers around here. So then 6 seconds, one of them comes on the screen. We do that again, we don't see any significant difference between the two groups. Again, you can't really differentiate the group as a whole if you have imagery or no imagery. So in terms of performance and capacity, things look very similar with or without imagery. Again, really interesting. However, if you start looking under the surface, looking under the hood, and ask people, how are they doing these memory tasks? Right? What's their strategy? What do they do to try and remember the shapes or the numbers? You start getting very different answers. People with imagery will say, yeah, well, I just visualized, 3 of those 3 as patterns, the different orientations. I could hold that image there in my mind on the screen. No problem. What are you talking about? Someone without imagery is not going to answer like that. They answer very differently. They're using a very different non-visual strategy. They're using geometry. They're using spatial locations. They're using symbols, all kinds of different things, different kinds of clustering and grouping and combinations of the things I'm describing. So once you start asking these questions, things are quite different. It's only when you look at overall performance, things look about the same. And there's some other interesting details there that suggest or provide more objective evidence that those with imagery are using a more low-level sensory method to remember things compared to those with aphantasia. So that's short-term working memory. So we're also really interested in episodic memory, so episodes of my life, not facts or semantics, but episodes, things that I lived in the past. What is my memory like with or without imagery? So here's some data based on a bunch of different questionnaires. Where the stars there, we're seeing significant differences between those with imagery and those who have aphantasia across these different measures of episodic memory, if you like. And there's parts of episodic memory where people tend to get you to try and come up with a future scenario, something in the future. They still call it episodic memory, but it's really episodic future thinking or planning hypotheticals in the future. So again, they're questionnaires. We want to try and get a more objective peek at this. So we use something that's sort of what's called an autobiographical interview, and it's a slightly adapted version of that. So where people are asked to come up with 6 different memories. So real memories that happened to them in their past, and also come up with 6 hypothetical future events that might happen to them, right? And these are all sort of within bandwidths of different distances from now in the past into the future, right? And then the people doing the experiment have to go describe in as much detail as they can these memories or these future potential future scenarios. So here's some data just to give you a taste of this. Again, we're looking at people with aphantasia and blue controls with imagery. And this is just a tally, if you like, the count of the number of details in the different conditions. So in the past here, you're seeing a lot more of, not huge, but significantly more details if you have imagery compared to not having imagery. Likewise, in the future scenarios, you're seeing more details in those with imagery compared to those without imagery. And you can map that out if you wanted to over the different time points. But I think this is a real take-home message, that there does seem to be objective differences in the amount of detail that those with imagery can recall. In other words, imagery, bringing an image to mind, gives you extra details for free, would be the way that some people might want to phrase that. Right, so personal details of your life seem to be different with or without aphantasia. And I want to make a clear point here that these are not catastrophic differences, right? It's not like you can't remember anything, because you clearly can. I know there's lots of individuals with aphantasia listening to this talk, and you clearly have episodic memory. So I'm not saying that. I'm just saying on the whole, when you look at the groups of people, there are significantly less details, right? So there are clear differences, but the memories are still there, absolutely. They just seem to be stored or accessed differently, and something is a bit different with those memories, right? They're not in that pictorial format that they would be if you have imagery. And that seems to affect the amount of details in episodic memory. So I want to switch to something else now, and this is something called mental rotation. Some of you in academia would have heard of that. So this dates back to a paper in the magazine journal Science, right, one of the top scientific journals, in 1971. And it was sort of a lot of this breakthrough in mental imagery research. It was sort of considered the first objective, reliable way to measure imagery, kind of like how I'm talking about that binocular rivalry technique at the beginning. And you'll notice I didn't mention this at the outset, and it'll become apparent why, or why not. So here's how it goes. You get given these different three-dimensional block things, and there's 2 of them, and you have to say whether the same block, one's just been rotated a different amount, or they're different. That's it. Seems pretty easy. And what's interesting when you do this task, the more those 2 things have been rotated away from each other, the longer it takes you to answer. You get this really nice linear response. This data is almost too perfect, too good to be true, right? And this was taken as, oh wow, this is evidence that people are rotating one of the objects in their mind's eye, right? This beautiful linear relationship. And it's literally that the time it takes is linearly tapping into your mental rotation of the object, right? And in hundreds and hundreds of papers, this has been used as a measure of mental imagery, right? So it's been around for, like 50 years. Right, so it's a classic measure of so-called mental imagery. So we ran this task with the blocks and a couple of other different versions of this task. There's all kinds of different things you can rotate, little mannequins, little people. So I'm just going to show the data from the blocks here, right? Again, in people with imagery, people without imagery. And here's the data here. Red in aphantasia in red and controls in the white there, and the individual data plots are blurred over the top there. And here's what's really important here. You see this linear relationship, this nice linear relationship that it's taking longer to answer the greater the rotation in the blocks, but you're seeing it in both groups. You're clearly seeing it in aphantasia, right? You're also clearly seeing it in those with imagery. And we see this, we've replicated this a number of times and across different tasks and different things, you tend to keep seeing this in both groups. So, right, we can interpret this in a number of different ways. One thing is what we tend to call mental imagery, visual imagery, is I don't think what this mental rotation task is measuring. It's measuring something else. The way our brain processes information, it comes in visual information through our eyes, and somewhere before it gets to all the way through our visual cortex, the information tends to get split up. And a lot of it travels up the top here, what's called this dorsal stream areas, which tend, in general, to process more moving things, spatial locations, spatial coordinates. And all the data we're seeing from those with aphantasia is that these spatial coordinates are just fine. And that's what I think this task is tapping into, spatial transformation. You don't need the imagery of the blocks per se, right? The blocks are kind of nothing. It's the spatial transformation. So it seems that even if your imagery, your object imagery to create the object of an apple, right, is not there, your spatial awareness, your spatial location, your spatial coordinate system is still there, and you can do these mental transformations. Right, so I think we should reclassify what this mental rotation task is, because it's not measuring imagery per se, it's measuring spatial coordination, spatial coordinates in space. So we ran another experiment looking at what I'm going to call thought control. So we've been fascinated by thoughts, how we control our own thoughts for a long time in the lab here. All kinds of, it's just fascinating, right? The idea that I can hold my hand out, arm out like this still, right? And I can do that for much longer than I can in my own mind. In other words, I can't keep my mind quiet and still for anywhere near the amount of time I can, right? And I don't do daily meditation or anything like that, so I don't practice this a lot, but I have a lot of trouble keeping my mind focused and still or empty. And I find that really fascinating. So here's the kind of task I'm talking about. The word horse flashes on the screen, right? And then it says, don't think about the horse, right? That's your only task. Do whatever you want, but don't think about the horse. You have 10 seconds of waiting and trying not to think about the horse. And if that horse, or whatever it might be on that trial, pops into your head, you hit the button, right? That thought intrudes, it pops into your mind, you hit a button and report that. Okay? If it does, then afterwards you tell us, was it an image, was it a sound, was it a smell, was it something else, abstract? And then we have CAS trials and other things like that. So again, we ran this kind of task with those with imagery, those without imagery, and we see a significant difference, a clear difference in that those with imagery have overall more thought intrusions. This is just the number of intrusions. Those, it's a bit hard to see here because we're showing all the individual data, but those with that are aphantasic have a lower, on average, number of intrusions. They're just able to keep those objects out of their mind, and we're also timing how long people hold the button down for. And if you look at the timing data, the button is down much longer in the control group as well. Right? We can also break things up. And as you might expect, if we look at the types of thought intrusion, what is jumping and popping into people's minds, we see the big difference there with images, right? People with imagery, pretty much a large bar there. It's mostly images, of course, not with aphantasia. It's mostly abstract concepts and things. So we're seeing a clear difference there and a clear difference in the number of intrusions. Right? Oh, you can look at that in a number of different ways, pie charts and different things. The idea is that not only is it a different amount of intrusions, the content and the characteristics of those intrusions are different with and without imagery. Right? So we're running more experiments to this at the moment, trying to use our binocular rivalry techniques to try and tap into this. But it seems so far that those with aphantasia might have better thought control. They might be able to keep things out of their mind when they want to, right? So in other words, if thoughts take on the format of an image, they're harder to keep out of your mind. And again, really interesting, really fascinating implications. So before I finish up, I wanted to sort of just, because this conference has got a lot of artists, they have the art exhibition, and a lot of people are asking about creativity and aphantasia, and there's this thing that without mental imagery or without imagination, as some people might say, you can't be creative. And so I wanted to sort of show a little bit of a taste of some new data we've been getting from experiments looking into this. And it's a topic that I've been wanting to study for many years, right? I went to art school. I love painting. I love drawing. I used to do these sort of in detail surreal drawings before I went on to study filmmaking at art school, before I did a PhD and went into neuroscience. So this is something that's really dear to me. So creativity, right? It's a hard thing to define. There's lots of different ways of defining it, and lots of people define it in very different ways. It's obviously important. It's going to be a big deal as we move into the future, right? And things become more and more automated. AI takes over more and more, right? You have World Economic Forum saying creativity is one of these top 10 skills of future work and future education, right? They call it a soft skill, terrible name. It's a hard skill, right? It's the hardest thing to measure, the hardest thing to understand. It's a hard skill. Let's not call it a soft skill. So it's a big deal, something very important. It hasn't always historically been measured that well. So what is it? We can talk about it in terms of painting and drawing and things like that, the classic art school things. That's not the way I think about creativity. I think whether you're dealing with numbers, an accountant, whether you're painting or being a sculptor, or you're an architect, or you're a vet or a medical doctor, whatever it is, there's a role for creativity. You can be creative. You can come up with novel ways of doing things that have impact. And that's kind of the essence of creativity, coming up with something novel that's also useful and therefore can have impact. So we ran an experiment using one of the classic ways of looking at creativity. It's called the alternative use task. And you get things like a brick, a paper clip, a bit of paper, and you're told you have a time limit and you have to write down all the different uses of that object that you can possibly think of. Right? So a brick might be build a house, smash a window, be a paperweight. And then, you come up with 10, 20 of these things, and it starts to get harder and harder. So then you can rate these responses in a number of different ways. So here's some data here. The turquoise there is those with aphantasia, and this dark purple is controls with imagery. So let me unpack this before we get too crazy excited by this. We're showing flexibility, detail, fluency, the number of different words, and how original they are, rated by multiple people. And yes, what we're seeing in this data is actually in 3 of these subclasses, those with aphantasia are performing better. They're ranking more highly. They're ranking more highly on fluency, on originality, and flexibility. Now, these aren't massive effects, they're only slightly better in aphantasia. But I want to sort of throw out a caveat here, or something that I think we haven't controlled for in these data, even though they are really interesting. Those who have aphantasia on our database have come to us. They've heard about aphantasia, they've read about it, they've got in contact through one way or another. So those people are the self-selecting bias. They're going to self-select by the nature that you if you are aphantasic, have heard about it, read New York Times article, read one of the different New Scientist things. You've made an effort to find out about it, understand it, apply, come and do the experiment, right? And that is a selection, we call a selection bias in psychology. So far with these data, we haven't got the group of hyperphantasics here, which would help control for that. So I think some of the better performance here, if you like, for those with aphantasia might be to do with that selection bias, but we don't know yet is the take home. What I want to get you to understand from these data is that at the very least, there's no decrement here. So even if this difference, this slightly better performance for aphantasics is due to response bias, there's no decrement here. So the emails I get every week saying that I can't be creative because I don't have mental imagery or visual imagination just don't seem to come true in this task. Even if you take out that self-selection bias, I don't think we're going to see people with aphantasia doing worse at these classic creativity measures. And that should be a comfort if you have aphantasia. Just quickly, one more test we did. It's called the remote associates test, where you get 3 different words and you have to say what is the common word. And this boat links those 3. Ran that aphantasia and controls. Again, we see a significant difference. Aphantasics are doing better again. And again, I think we have to be careful with how we interpret this until we have the hyperphantasic data to make that comparison. So at the very least, I think it's exciting that you can take these data, even if it turns out to be, these 2 groups tend to be effectively the same, there's no decrement without mental imagery. Without visualization capacity, you're not any less creative, right? You can still do the things you can do. You can't use your lack of visual imagery as an excuse to not be creative, in other words, right? And we know this from all the artists. We're seeing this from Ed Catmull and his top animator in Pixar, and all the entrepreneurs I've talked to who are aphantasic, right? I don't think there's any evidence that's going to pan out that there's any differences in creativity without mental imagery. So let's just wrap things up now. So we can measure imagery objectively and reliably, right? I gave you at least those 3 different ways we're doing it in the lab: binocular rivalry, emotional response, and the pupil response, right? We now can do this. We have this blood test for the imagination, for the mental, for the mind, this objective microscope. We can measure these things. So what is imagery? How can we bring all this back together? How can we think about imagery, right? If you have it, you tend to use it for a whole lot of things. You tend to use it for navigation, for memory, for all kinds of things. If you don't have it, you can still do most of those things pretty much the same, right? There are a few things we're seeing with emotional response, with episodic memory, where there are some differences, but mostly we're not seeing differences now. So how can we think about imagery? Well, it's a tool that if you have it, you'll tend to use it. If you don't have it, you'll find a different tool or tools to get the job done. I think that's the best way to think about imagery for now. It's a simulation engine like virtual reality, which lets us conjure up pictorially things that are not here right now. Lets us break away from here and now in time and space. But you can achieve those goals in a very different way, but you can still do them without mental imagery. Right? So as I said, it's at once ubiquitous, but it's not necessary. We can do it in a number of different ways. And for me, imagery is really the key to our private internal worlds, right? It's the format, the characteristic, the nature of our thoughts, literally the format of our thoughts. Are they pictures or are they something else? Are they symbols, words, something else, right? It's the very nature of who we are if our thoughts make up who we are. And I think it's an amazing way to study consciousness, internal personal consciousness. And when I started out studying imagery 13, 14, 15 years ago, many said it was just too hard to do. You can't measure it, you can't study it objectively and reliably, why would you do it, right? A couple of people even said, don't do that, Joel, it's career suicide. How can you study something if you can't measure it objectively and reliably in the mind? And, right, as you've probably guessed, I don't agree with that. I think it's the exact opposite. I think us as scientists now have a responsibility to move towards those things. If we don't have a way to measure it, right? If the way we're measuring it is inadequate, then more people need to study it, not less, right? More people need to study intuition, need to study hallucinations, need to study creativity, need to study resilience, need to study, you name it, the fluffy thing in the mind that has impact to our lives, but it's difficult for neuroscience to measure. More people need to go there. More people need to study it. So that's my sales pitch for a career in mental imagery. I want to thank a bunch of people from the lab who have all differentially contributed to the data from today's talk. It's not just me. Rebecca Keogh, who is talking tomorrow, had a hand in most of the things you've seen in my talk. Lachlan Kay, Alexi Dawes, Marcus Wicken, Mary Anthony Godwin, and Belinda O'Brien all had their hand in different experiments in different ways. So thank you to them. And thank you to you guys who have, if you listen to this, taken part in some of our research. There's so many questions, there's so much research we want to do, and we couldn't, of course, do it without you, so thank you. I also need to thank my university, the Australian government agency which fund our research, and of course, thank you for listening. So thank you. Right, we're Future Minds Lab. My name is Joel Pearson. If you're interested in our research, find out more, go to Future Minds Lab. You can just Google it, but you can also type this in, /extreme imagery. And you can see there, you can sign up to be a part of our database, see some of our papers, watch some other talks I've done. Yeah, hope you enjoyed it.